have another software demo. So I'm told no Q&A, but you can find Deep Ganguli at the poster break. The demo, he's from Chang Zuckerberg Initiative. What's he got a name for the demo? A comparative and unifying approach to image-based transcriptomic data analysis. Thanks.
Thanks, Lydia. I'm mildly embarrassed to say that I will not be talking about deep learning today, despite my name being Deep. Also find it somewhat amusing. Rather, what I've been working on more recently is on algorithms and tools for the Human Cell Atlas project. Roughly, the goal of this project is to profile the order of 30 trillion cells in the human body across multiple humans. There's two main branches, a cellular branch and a spatial branch. For now, this is roughly an exercise in RNA profiling, although not exclusively. And on the cellular branch, people are using a lot of single-cell RNA sequencing experiments, which are great, except the problem is the cells are dissociated from their native tissues before the RNA is profiled. And of course, spatial information has a lot to do with cell identity and what these cells are actually doing. And so on the spatial branch here is what I'm going to be focusing on. Looking back, as we're tool developers, for the sequencing-based side of things, there's been a lot of work on sort of standardized processing pipelines and file formats. And there's a lot of really good existing tools for getting out what, at the end of the day, is a cell-by-gene expression matrix from which one can do all of the biology that they're interested in. On the imaging-based side of things, we get different kinds of data, of course, images off of a microscope that come in all shapes and sizes and file formats. And there's a relative paucity of good tools that go from these images into the cell-by-gene expression matrix. And of course, here we additionally have information about where the cells are in space and their morphology and where the genes are within the cells as well. So for these assays, there's at least 10 of them. And we've been looking at data from all of these assays, so things like marefish and basic SM fish and seqfish and so on and so forth. And so we kind of refer to this project as starfish, a little programming joke. I got one laugh out of that. That's great. And so we're working with this consortium headed by Ed Lean here at the Allen Institute for Brain Science, and my close collaborator Brian Long, and many, many others that are involved in this project. And it's a consortium effort to compare and contrast all of these different methods for differentiating cell types in the human brain. And the intuition for what these methods do is that you design your probes a priori for the RNA transcripts you're interested in imaging. Then you hybridize your probes on, you image, and in the most basic way of doing this is that you get the number of genes you can profile with one round of imaging is the number of fluorophores you have available to yourself. So that would be something like SM fish. And to get more genes out of that, you strip your probes, reassign which fluorophores correspond to which genes, and then re-image. And so that scales your number of genes is equal to your number of rounds of imaging times your number of color channels that you have available. It's going to be hard to get transcriptome-wide. Many of these other methods multiplex by assigning different patterns of fluorescence activity across many rounds and many channels to different genes. And these things scale exponentially in your number of rounds of imaging, which is really nice. And then there's other methods which are, for example, in situ sequencing is literally sequencing by litigation in situ, where you're actually reading off the base pairs of the mRNA molecules. So all these methods vary in sort of the encoding scheme and the chemistry that they're using. And what does the data analysis look like? The data analysis looks, roughly speaking, like this. So you have your images come in, and they ought to be registered across rounds of imaging such that the molecules you're detecting are not shifting too much in space. And then there's some filtering done to kind of remove the background autofluorescence and enhance the actual spots. And the spots correspond to the RNA molecules. And so those spots have to be found, which is a non-trivial problem, and then decoded such that you can attribute a different identity to each spot, which corresponds to its gene, shown here in pseudocolor. And then finally, the cells have to be segmented. This was my chance to talk about deep learning, and assigned to the actual cells. And what we saw was that if there's n assays, there's also n analysis pipelines. And It's actually pretty amazing. I was allowed to kind of go in and look at how everyone's doing it, and everyone's doing really, really cool stuff. And everyone's using really different toolkits, software packages, different inputs, different outputs. And so we thought maybe we could help. We had a hypothesis that maybe we could help that hasn't been disproven yet. And so we call this Starfish, and really what it is, it's just a library where we can sort of share knowledge for about how we want to process these data. So it's not a pipeline, it's not an N+1 analysis pipeline per se, but rather it's a library that everyone can use and contribute to process these data. And our goal here is just to sort of enable and accelerate progress in this area. And it's a team effort. We were a group, a mix of traditional software engineers and computational scientists. So in terms of like what are the inputs and outputs to these things, we've tried our best to define standardized inputs. It's really not rocket science. The inputs are just single plane TIFFs with JSON metadata that tells you which round of imaging and which color channel you're looking at. A codebook is simply a JSON file which has for each round and for each channel some elements you expect to be on in your image. So this is the code word for example, one gene. I would expect this pattern of fluorescence activity across my rounds and channels. And then I have a list of these for everything else. It's my code book. And finally, our standardized outputs are all the things you'd expect. For each spot, where is it in space? For each cell, what does it look like? What's the boundary? And what we've shown so far is at the very least, we can show that we can reproduce the results of other pipelines with this one library. So for example, here we're looking at, or we have looked at, for example, 5 different assays. We've looked at many more, and they all take different steps through the Starfish library. So although I said filtering, there's many ways to filter. I'm just showing 2 here. Finding spots, I'm showing 2 here, but there's many ways in the library. Decoding actually are only 2 ways in the library. And then segmentation, which I'll touch on in a little bit, and finally getting out expression counts. So in this tube map, you're sort of free to go off and kind of take your own journey through how to process these data. But since we've been able to standardize on the inputs and like the intermediate file outputs, we can really experiment with sensitivity analyses. Like what really matters? Is there one way to process these data? Or is it truly that there's n ways to do it? So it's sort of an open question. Preliminary evidence suggests that there's less than n ways to do it, but not 1. Which maybe it had to come out that way. So does it work? Yeah, don't worry about it. It works. Here's some plots. I can kind of get into that, but I won't. The other cool thing you can do are these interesting analyses of the data. So for example, here, recall that like when we're finding our spots, we're measuring their intensity values across rounds and channels. So you can imagine for each, I have a matrix where each row is a spot and each column is the fluorescent is an actual index into a round or a channel, and each element in that matrix is the intensity of that spot. And now for all of those spots, I've also decoded them. So I know what those genes correspond to. So when I have a matrix, I can project it into 2 dimensions. So here are t-SNE plots of those pixel traces or those spot traces across 3 different assays, color-coded by the gene identities. So what this tells you is, for example, for in situ sequencing, all these green spots correspond to some gene, and so do these purple spots, and there's some nice separability in this low-dimensional representation of this data, which means that it gives you sort of a sense of the SNR, like what is my ability to actually process these data to decode what the gene identities were. And in Marefish, for example, these pop out as nice Hamming spheres, which is sort of how they designed their codebook a priori, such that like a gene was expressed as a unit sort of sphere in this high-dimensional space. When you reduce the dimensionality, that sort of pops out at you. And so for Dartfish, for example, yeah, this could be, it's sort of a visual diagnostic to go back. So is it something with the encoding? Is it something with the way that the data were processed? It's sort of unclear, but the nice thing is we can do these comparative analyses and ask questions about how to sort of benchmark the signal-to-noise ratios and the decoding capacities and so on and so forth. Here's, it's a library, so it's very boring to give these demos since it's just code, but here's an example of some code. Wow. So, right? So it's, load data, do all of your stuff with it. The thing I want to impress upon you is that if you switch to the Marefish pipeline, for example, same basic things, some different decisions, but we put a lot of work into making the API Pythonic and usable. So it's really, analyzing the data from an experiment should not be, piles and piles and piles of MATLAB code. It can be like sort of short snippets. And so, right, I sort of was alluding to this earlier. There's many options for each step. Whenever I talk about starfish, everyone's very interested in, well, how are you segmenting cells? And the answer is there's, right now there's only one option, which is a seeded watershed segmentation that's actually implemented in starfish. And it's a very domain-specific application of a known segmentation algorithm. But we're also experimenting with other ways for doing this. So we have an API contract in a branch that will take your pre-trained machine learning model. Here I'm showing it taking in a pre-trained elastic model and then showing the results of the segmentations. And we'll also take in your Fiji ROI sets and use those to segment the data as well. And as you can see, this is a really hard problem. There's a lot of heterogeneity in how these things work. So here, for example, here it's segmented right. There it over-segmented, and then the manual annotation is, of course, different than all of it. So hard problem, and that error will propagate to the subsequent cell-by-gene expression matrices. So there's an interesting statistics problem hidden in there. And then finally, I want to give you a worked example. So we took an SM fish data set from our collaborator here at the Allen Institute for Brain Science, 75 fields of view, 3 color channels, 9 rounds of imaging, lets you resolve 27 genes, processed it through starfish. Here's a plot of all of kind of the spots color-coded by the gene identities. And I should say that this sample is from Cortex. And a neuroanatomist did a sort of heroic job of actually manually segmenting all of these cells as well. So now we can attribute each spot in here to each cell to make a cell-by-gene expression matrix. And now we can actually do some biology. And so here's 2 ways of looking at that final data. On the left here, we have for each cell is a point. And we have their position in space. X is on the x-axis and y is on the y-axis. And the spots have been color-coded by which layer that they belong to in the cortex, 1 through 6, as done by the neuroanatomist. And here's another way of looking at that data. So we take that cell-by-gene expression matrix, which for each cell you have a vector that's 27-dimensional. We project it down to 2 dimensions using a UMAP algorithm. And again, we color code them by which layer they belong to. So cells that are nearby in this UMAP space also have similar gene expression patterns. And you can see here that, so for example, these layer 2, 3 cells in red, yes, also form their nice cluster in gene expression. So they're similar according to both a neuroanatomist kind of looking at morphology, but also in terms of their molecular properties. And I can also explain away all the funniness in here too, if anyone wants to talk about that later. And then here's another way of looking at it. So we're looking at the expression levels of a gene, CUX2, which is a marker for layer 2, 3, and 4. And we can see it's indeed highly expressed in layers 2, 3, and 4. And also over here, these were the sort of areas of that UMAP plot that also correspond to those layers. So it's just an example, a fully worked example of taking raw data and getting out some biological analysis using the starfish package. And so I guess you can come to the demo. We can kind of do whatever you want. Thank you.